This is a chemical known as dimethyl sulfoxide, or the MSO as an abbreviation. The MSO is a colorless solvent and is used to dissolve both polar and non-polar compounds. In medicine, it is used as a treatment of interstitial cystitis, leakage of intravenous drugs from vein into surrounding tissue, or known as extravasation. It is easily absorbed by the skin and used in conditions such as scleroderma and scars. In laboratory practice, the MSO is used to dissolve a drug compound and also for cryo-freezing media or cryoprotectant. Normally, we add 10% of the MSO at the concentration of 100% into our working media, and this will prevent intracellular and extracellular crystal from forming in cells during the freezing process. This crystal can cause cell death and can't be used for transplant or cell culture work. In another condition, the MSO is used in banking of cord blood cells where 10% of the MSO is combined with saline or albumin. This is a picture of fetal bovine serum, or abbreviated as FBS. Now let's learn what is FBS. FBS is basically liquid fraction of clotted blood from fetal calves. It does not contain cells, fibrin, and clotting factors, but it contains a large number of nutrition essential for the growth of the cells in cell culture work. Bovine serum albumin is the major component. It contains attachment factors, growth factors, hormones, lipids, spreading factors, sugar, transport proteins, and vitamins. You might be wondering why fetal bovine is used. Well, fetal bovine contains sparse amounts of gamma globulin and fewer complement proteins. So this makes FBS ideal for propagating cells, for growth, and reduce the possibility of mammalian cell binding or lysing in the culture. So FBS should be stored at minus 20 degrees Celsius and thawed between 2 to 8 degrees before use. Next picture is trypan blue. Trypan blue is a stain to assess the cell viability, and it is used in dye exclusion tests. It is used to count cells with hemocytometer during subculturings. Live cells do not take up trypan blue, but dead cells do. How is it possible? This is based on living cells resist permeation by the chemical stains. However, in dead cells, there will be broken cell membrane which allows or permit the trypan blue to enter the cells. This picture shows HPase. HPase is basically a buffer. It is incorporated into medium to stabilize the pH of medium, as open dish may cause pH to rise, while overproduction of carbon dioxide and lactic acid produced by the cells may cause the pH to fall. How to keep HPase? It needs to be kept between 2 to 8 degrees Celsius and ensure that the content is not contaminated each time you open the bottle. This picture shows the antibiotics penicillin and streptomycin. Most of the students call it pen and straps. These are the antibiotics that commonly used in cell culture to prevent bacterial contamination against gram-positive and gram-negative bacteria. The two antibiotics usually comes as a combined antibiotics in one bottle. The solution contains 5,000 units per ml of penicillin and 5,000 microgram per ml of streptomycin. When preparing a complete media, we use 1% of penicillin and streptomycin from total volume during preparation. Penicillin inhibits cell wall synthesis of bacteria, while streptomycin interferes with protein synthesis of bacteria. The next picture shows trypsin EDTA. So what is trypsin EDTA? Trypsin is basically a solution consisting of single-chain polypeptide of 223 amino acid residues. Trypsin is used to remove adherent cells from the culture surface in the flask or wells or dish plates. It is normally kept between -10 to -40 degrees for long storage. However, trypsin can also be kept at 4 degrees. We discourage the practice of a repeated cycle of freezing and thawing, so you can aliquot small volume of trypsin and store at 4 degrees for daily use. The optimal pH for trypsin activity is 7 to 9. EDTA function as a chelating agent that neutralizes the calcium and magnesium ions that obscure the peptide bonds. The presence of serine protease inhibitors such as aprotinin might inhibit the trypsin activity. This picture shows a container containing 70% alcohol. 70% alcohol is used in cell culture studies as it has been found to be the most effective concentration in killing microorganisms. Why not using 90% or absolute alcohol? Well, the higher percentage of alcohol will make the solution evaporate faster, therefore is not effective in killing microorganisms. The pure alcohol can denature the protein in contact in all directions. The denatured protein would then stop the alcohol from penetrating further inside the bacterial cell, and this leads to inactive but not dead. When the condition permits, the microbes will be alive again and will start functioning and propagating. So when we use 70% alcohol, this concentration can still denature the protein but at a slower rate. Therefore, the penetration can occur through the cell before coagulation or denaturation process takes place. Then, the entire cells will become denatured and cause microbial death. This is a picture of phosphate buffer saline, or abbreviated as PBS. PBS is basically a buffer solution commonly used in biological research. It contains disodium hydrogen phosphate, sodium chloride, and it is water-based. The buffer helps to maintain a constant pH. The osmolality and ion concentration of solutions are almost the same as human body. PBS is used for rinsing the cells in cell culture, and as well as it can be used as virus transport media. It comes in a tablet form as well as a ready-to-use solution. For the tablet form, it needs to be dissolved in ultra-pure water and autoclaved before it can be used in cell culture to maintain sterility. The next picture shows the growth factors and hormones. In cell culture, some cells require growth factors and hormones to stimulate cell proliferation. The EGF and insulin, for example, stimulate cell proliferation by promoting glucose and amino acid uptake by the cells. Apart from growth factors and hormones, vitamins are also used as a maintenance of healthy cells and as well as antioxidants. Example of vitamins that are used in cell culture work includes ascorbic acid and the B group's vitamins, which are important as a growth stimulant in certain cells.